They are not viruses currently circulating through your blood. Most are damaged, fragmented, chemically silenced, and incapable of producing a complete infectious virus. Yet their ruins still contain switches, promoters, binding sites, proteins, and regulatory instructions that evolution has repeatedly learned to reuse. The popular version of this story says prehistoric viruses hijacked our ancestors and built the modern prefrontal cortex. The real story is more complex and more astonishing. Viruses did not plant intelligence. Natural selection salvaged useful parts from countless genomic invasions. To understand how that happened, we must return to a time before humans, before apes. Retroviruses were moving through ancient populations, searching for cells they could enter, exploit, and convert into factories for their own reproduction. A retrovirus normally carries its genetic information as RNA. After entering a cell, it uses reverse transcriptase to create a DNA copy, then uses integrase to insert that DNA into the host chromosome. Once embedded, the viral sequence can command the cell to produce more viral material. Most infections end with the infected individual, but occasionally a retrovirus reaches an egg cell, a sperm cell, or a cell that will produce them. If that infected individual survives and reproduces, the viral insertion can be inherited by the next generation like a genetic mutation. At that moment, an external virus becomes endogenous. It is no longer only an infection passing between bodies. It has entered the hereditary archive of a species. Every descendant may carry the insertion, whether or not the original virus still exists in the environment. Over evolutionary time, most such insertions decay. Mutations break viral genes. Deletions remove essential segments. Host defenses wrap the region in repressive chromatin. What remains is often a fossil, recognizable enough to reveal its origin, but too damaged to recreate the ancient pathogen. Yet evolution does not judge DNA by its history, it judges consequences. If a viral sequence damages survival or reproduction, selection tends to remove it. If it is neutral, it may drift. And if one fragment accidentally improves development, immunity, or brain function, selection can preserve it. This process is called co-option or domestication. A structure evolved for one purpose is recruited for another. Viral genes may begin as tools of infection, and later become switches, construction materials, or communication systems inside the host. The human genome, therefore, is not a clean blueprint written from nothing. It is a layered manuscript, edited by mutation, duplication, recombination, disease, and conflict. Ancient viruses did not simply leave scars in that manuscript. Some supplied sentences that later editors refused to erase. The most visible example is not the brain, but the placenta. Mammals have repeatedly domesticated retroviral envelope genes, which originally helped viruses fuse with host cell membranes. In several lineages, related captured genes now help placental cells fuse and build the interface supporting fetal development. But the brain presents a special challenge. It is not built by one master gene. Human cognition emerges from thousands of genes, regulatory regions, developmental timing systems, cell interactions, environmental inputs, and evolutionary trade-offs. No single viral insertion created language, self-awareness, planning, or abstract thought. Instead, ancient viral material appears to have contributed to the regulatory landscape through which many ordinary genes are turned on and off. This distinction is crucial. The most important legacy may not be viral proteins themselves, but viral control sequences repurposed as switches near host genes. Retroviruses carry long terminal repeats, or LTRs, at their ends. These regions contain signals that once controlled when and where viral genes were transcribed. After integration, an LTR can sometimes influence nearby human genes, acting as a promoter, an enhancer, or a source of regulatory RNA. Imagine thousands of foreign switches being scattered across a genome. Most are disconnected, suppressed, or harmful. But occasionally one lands near a gene where its response to a transcription factor becomes useful. The insertion creates a new pattern of gene activity that did not exist before. Over millions of years, this can help rewire developmental programs. A gene may begin turning on in a different tissue at a different embryonic stage or in response to a different signal. Small shifts in timing can have large effects when the tissue under construction is the cerebral cortex. The cortex develops through carefully ordered waves. Neural progenitor cells divide, adopt identities, migrate, extend axons, form synapses, and become organized into layers and regions. Each transition depends on genes activating at the correct intensity and then shutting down before the next developmental decision. Endogenous retroviral sequences are embedded within this regulatory environment. Studies of developing human brain tissue and neural progenitor cells have found that their activity is neither uniform nor random. Different families and loci show region-specific and stage-specific patterns, suggesting they are woven into developmental control systems. One key guardian of this system is TRIM28, a protein that helps silence transposable elements by recruiting machinery that compacts chromatin. In human neural progenitor cells, TRIM28 does more than suppress genomic parasites. By repressing particular endogenous retroviruses, it also affects nearby genes and regulatory networks. This creates an evolutionary paradox. The host must silence ancient viral elements because uncontrolled activation can be dangerous. But the same silencing machinery has become part of normal gene regulation. The genome is not simply fighting an enemy, it is managing inherited technology that is useful only under strict supervision. During early development, that supervision becomes especially delicate. Soon after fertilization, much of the embryo's epigenetic landscape is reset. Chemical marks are erased and rebuilt. The genome enters a temporary state in which regions normally locked down may become accessible. This is when certain endogenous retroviruses awaken in highly specific patterns. HER-VK, one of the youngest and best preserved human retroviral families, is transcribed during normal pre-implantation development. Its activity begins around embryonic genome activation and continues into stages when the earliest cell lineages are being established. Researchers have detected HER-VK proteins and virus-like particles in human blastocysts. That does not mean the embryo is suffering a conventional infection. It means early human development unfolds in the presence of molecular products inherited from infections that occurred in distant primate ancestors. Even more surprising, one HER-VK protein called REC can interact with cellular RNA and may stimulate antiviral restriction pathways. In experimental systems, its expression increased to surface protein that can make cells harder for some modern viruses to infect. An ancient invader may therefore help prepare embryonic defenses. The relationship, however, is not simply beneficial. Too much HER-VK activity can disrupt cortical development in laboratory models derived from human stem cells. Elevated transcription altered neuronal differentiation and cortical patterning, partly through changes involving the receptor NTRK3. This is the recurring theme. Dosage, timing, and location determine whether the viral legacy is useful or destructive. A sequence can support one developmental state, disturb another, and become dangerous if reactivated later in life. Evolution rarely produces perfect components. It produces systems balanced around acceptable risk. Another family, HER-VH, is deeply connected to human pluripotency. Pluripotent cells can still become many different cell types, including the cells that eventually form the nervous system. HER-VH sequences are strongly expressed in human embryonic stem cells and help support the regulatory network maintaining that flexible identity. Some HER-VH copies function as long non-coding RNAs. Others provide binding sites for transcription factors or contribute to boundaries in the three-dimensional organization of chromosomes. These roles influence which genes can communicate across folded regions of the genome and which developmental programs remain available. This does not mean HER-VH alone made the human brain possible. It means primate evolution incorporated retroviral material into the circuitry controlling human stem cell identity. Remove or suppress certain HER-VH transcripts experimentally, and pluripotent cells can lose aspects of self-renewal and begin differentiating. The brain's viral inheritance also extends beyond endogenous retroviruses in the narrow sense. Other retrotransposons, including line 1 elements, descended from mobile genetic systems that copy themselves through RNA intermediates. They occupy a vast portion of the genome and can influence neuronal gene expression and diversity. Line 1 activity in neural cells has generated intense debate. Some studies suggest that new insertions can occur in somatic brain cells, potentially creating genomic mosaicism in which neighboring neurons do not carry perfectly identical DNA. The scale, frequency, and functional importance of this process remain unresolved. What is clearer is that line 1 derived transcripts contribute to the complexity of the human neuronal transcriptome. In adult neurons, pieces of these elements can become incorporated into RNA molecules, creating primate and human-specific transcript patterns that may alter how genes are regulated or translated. Again, the lesson is not that uncontrolled jumping genes are good. Excessive activity can damage DNA, trigger immune responses, and contribute to disease. The important insight is that the nervous system evolved while surrounded by mobile elements and sometimes recruited their sequences into normal molecular routines. Then there is ARC, the discovery that makes this entire history feel almost tangible. ARC is an immediate early gene activated by neuronal activity. It is required for forms of synaptic plasticity, the capacity of connections between neurons to strengthen, weaken, and reorganize during learning. The ARC protein can assemble into capsid-like structures resembling the shells retroviruses use to protect their genetic cargo. These structures can package ARC messenger RNA, leave neurons within extracellular vesicles, enter other cells, and deliver the RNA into their cytoplasm. A mechanism once associated with genomic parasites has been repurposed as a communication system in the brain. The resemblance is not metaphorical. ARC evolved from a retrotransposon gag protein, the kind of structural protein that ancient mobile elements used to build protective particles around their own RNA. Why would a neuron need virus-like capsules? The exact physiological importance of intercellular ARC transfer is still being investigated. But neurons are unusually dependent on local, activity-sensitive communication. A capsid can protect RNA, concentrate cargo, and move information between cells across extracellular space. ARC also regulates receptors at synapses and helps stabilize memory-related changes. Its viral ancestry does not explain consciousness, but it demonstrates that at least one essential component of neuronal plasticity was assembled from ancient mobile element machinery. The mind uses a molecular descendant of genomic invasion to remodel itself. This discovery changes the emotional meaning of infection in evolution. We normally imagine a virus and host as separate opponents, but once viral DNA enters the germline and survives long enough, that boundary blurs. The enemy sequence can become a permanent substrate on which the host builds future adaptations. The transformation is not peaceful. For every useful domestication, countless insertions were probably harmful, neutral, or lost. Ancient epidemics may have killed individuals, reduced fertility, and driven population bottlenecks. The beneficial fragments visible today are survivors selected from an enormous history of biological damage. The prefrontal cortex, central to planning, inhibition, working memory, and flexible behavior, is especially tempting to place at the center of this story. Yet there is currently no evidence that one identifiable ancient viral infection directly constructed the modern human prefrontal cortex. What scientists can say is subtler. Viral-derived sequences contributed to genome regulation, embryonic cell states, neural progenitor programs, synaptic plasticity, and immune control. These systems all influence how a cortex develops and functions. Their combined effects may have shaped evolutionary possibilities available to primate brains. Human brain evolution often depended less on inventing entirely new protein-coding genes than on changing when existing genes were used. Humans and chimpanzees share most protein-coding sequences, but differ in developmental timing, gene regulation, cell proportions, connectivity, and prolonged maturation. Transposable elements are powerful sources of regulatory novelty because each family can distribute related binding sites across many genomic locations. When a host transcription factor begins recognizing those sites, an entire network of genes can potentially acquire coordinated responses in a relatively short evolutionary interval. The result is not a viral master plan, but a collection of evolutionary experiments. One insertion alters a nearby gene, another creates a non-coding RNA, another recruits chromatin proteins, another interferes with development and is eliminated. Over vast time, the surviving network can become inseparable from normal biology. The caution matters because repetitive DNA is technically difficult to study. Many Hervey copies are highly similar, making short sequencing reads hard to assign to a precise location. Older studies often measured entire families at once, obscuring which individual locus was active and whether it caused an effect. New long-read sequencing, single-cell transcriptomics, epigenomic maps, CRISPR editing, and brain organoids are changing that. Researchers can now identify specific viral-derived loci, measure their activity in particular cell types, disable them individually, and observe how developmental programs respond. Brain organoids are especially useful because they model aspects of early cortical development using human cells. They cannot reproduce a complete brain, consciousness, sensory experience, or full vascular and immune systems. But they allow scientists to test whether changing one Hervey locus alters neural differentiation or regional patterning. These models have already revealed the danger of oversimplification. A Hervey sequence may appear active during normal development, yet forced overexpression can produce abnormal outcomes. Conversely, global silencing may also disturb cell identity. Healthy development often depends on a narrow window between suppression and activation. That narrow window is maintained by epigenetic defenses. DNA methylation, histone modifications, Crabbe zinc finger proteins, TRIM28, CTDB1, and other systems recognize mobile elements and keep them under control. Many of these defenses evolve through an arms race with the elements they suppress. Every new viral invasion created pressure for better genomic surveillance. Hosts evolved proteins capable of recognizing particular sequences. Mobile elements mutated to escape. Hosts responded again. Over time, the regulatory machinery born from conflict became integrated into embryonic development and tissue-specific gene control. The brain may carry a particularly complex version of this compromise because neurons must remain functional for decades. They rarely divide, cannot easily be replaced, and are vulnerable to inflammation. A burst of retroelement activity that might be tolerable in another tissue can be catastrophic in a neural network. Experiments in mice show what happens when control fails during brain development. Removing TRIM28 from cortical neural progenitors caused strong endogenous retrovirus expression in descendant neurons. Later, the brains showed activated microglia and accumulations of retroviral proteins, linking developmental failure of silencing to lasting inflammation. Human studies have associated altered HRV expression with conditions including schizophrenia, bipolar disorder, amyotrophic lateral sclerosis, multiple sclerosis, and other neurological or psychiatric diseases. Association alone cannot establish whether HERV activity is a cause, a consequence, a biomarker, or some combination. Inflammation can activate endogenous retroviral sequences, while viral-derived RNA and proteins can themselves stimulate immune pathways. This creates possible feedback loops. Disease may loosen epigenetic control, awakened elements may increase cellular stress, and that stress may further destabilize regulation. In the developing brain, however, unnecessary antiviral signaling could interfere with proliferation, migration, synapse formation, or the behavior of microglia. The nervous system must distinguish useful expression of inherited viral sequences from activity that resembles a present infection. This connects ancient infection to a broader evolutionary principle. Immunity and brain development are not separate stories. The molecules used to detect pathogens often also guide growth, pruning, and communication. A defense system shaped by viruses can indirectly influence cognition even when no viral gene is domesticated. A mutation improving placental function can permit longer gestation or different fetal growth patterns. A regulatory change in pluripotent cells can alter when neural progenitors appear. A new enhancer can shift expression in one cortical layer. Evolution builds complexity through chains of indirect effects. The word parasite also becomes unstable across deep time. A sequence can begin as selfish DNA, become neutral after losing mobility, and later acquire a host function. Once the host depends on it, removing the sequence may become harmful. The former parasite has crossed into the definition of self. ARC is the clearest symbol of that crossing. A protein descended from a retro element now activates when neurons fire, assembles into capsids, and participates in the molecular events supporting memory. The brain did not merely survive viral-like machinery, it made that machinery part of remembering. HERVH offers a second symbol. Primate-specific retroviral sequences help maintain the flexible identity of human pluripotent cells and shape chromosomal organization. Before a single thought can exist, the developmental potential of the embryo is partly managed through regulatory material inherited from ancient infection. Together, these examples reveal no simple march from pathogen to intelligence. They reveal evolutionary bricolage, nature constructing new systems from whatever material survives. Viruses supply powerful modules because they already know how to enter cells, control transcription, package RNA, fuse membranes, and evade defenses. The cost is permanent vulnerability. Domesticated elements must be controlled. Their relatives scattered across the genome may still respond to stress, aging, mutation, or epigenetic failure. Evolution gains innovation, but inherits maintenance problems that can persist for millions of years. This trade-off may be unavoidable. A perfectly protected genome would resist invasion, but also lose a major source of novelty. A completely permissive genome would generate variation, but collapse under instability. Life evolves between those extremes, suppressing mobile elements while occasionally exploiting them. The human brain reflects that balance at extraordinary scale. It requires stability enough to preserve identity across a lifetime and plasticity enough to learn from experience. Ancient mobile elements contributed to both sides. Epigenetic systems that enforce silence and domesticated proteins that enable change. Even memory itself depends on controlled instability. Synapses must alter their strength. Genes must activate in response to experience. RNA and proteins must move to precise locations. Neural circuits must be remodeled without losing the organism's continuity. ARC's viral-derived capsid sits directly inside that paradox. When an experience activates neurons, ARC expression rises rapidly. The protein helps reorganize synaptic receptors and participates in consolidating changes across networks. Its evolutionary ancestor once served a selfish element. Its modern descendant now helps an animal convert experience into lasting biological structure. That is not proof that viruses created consciousness. Consciousness remains an emergent phenomenon whose biological basis is still incomplete. But it is proof that components of cognitive machinery can have origins radically different from the functions they now perform. The same principle applies across the genome. Evolution does not preserve labels such as viral, human, foreign, or native. Those categories matter in the present, but deep time cares only whether a sequence replicates and whether its consequences allow organisms carrying it to reproduce. An insertion that began as foreign DNA can eventually become older than the species carrying it. It may be shared across primates or mammals, inherited through every birth, and embedded in developmental programs. At that point, calling it foreign describes history, not current biological identity. Each surviving insertion also represents a chain of improbable events. A virus reached the germline. The host survived. Reproduction occurred. The insertion escaped elimination. Descendants carried it through environmental upheaval, competition, migration, and speciation. Later evolution found a way to use or tolerate it, multiply that chain across tens of thousands of fragments, and the genome becomes a battlefield archive. But the archive is not passive. Its old weapons have been dismantled, modified, and connected to new systems. The battlefield has been converted into infrastructure,